Good evening. I'm Serena West, and this is my thesis presentation on microbial communities of deep marine sediments. So deep marine sediments make up nearly 70% of the Earth's surface, so all that mud underneath oceans and seas. And they can go quite deep. There's ocean sediments that go down a couple of kilometers actually. So, but these environments are extremely difficult to access because they're underneath the ocean floor. So you need specialized drilling equipment that is ship-based. It has to drop through several kilometers of ocean water, and then when it hits the sea floor, it has to begin drilling and done in such a way as to preserve the integrity of the microbial communities and exclude contamination. So, but these sediments are really important because they're a time capsule, much like sea ice in Antarctica. These sediment layers build and they capture what is on the seafloor at the time of sedimentation, both living and non-living materials. So once an organism is in a layer of sediment, it's pretty much trapped there because pore size and it's really difficult to move. So, and these communities survive on extremely low energy, no oxygen, very cold, and like I said, very low mobility. So these conditions are very hard on the microorganisms that live there, but they are very good for preserving environmental DNA. So even if a microorganism dies in a sediment layer, there's the potential for the DNA to be preserved for potentially thousands of years and sampled later on. And so that is the core of this thesis work, is comparing the living DNA found in sediment microorganisms and the environmental DNA that was found in the same sample. So they're paired samples. So my research question is there a pattern of DNA degradation by uracil substitution within deep marine sediment microbial communities as depth increases? So I'll explain that a little bit. This is a sediment sample. This is a composite image of sediments as it comes out of a drilling hole. This particular hole is in the Bering Sea. And so the top row on the far right is, you see 0, that's the seafloor. And then everything to the left is going down, or sorry, switch that. Everything to the right is underneath the seafloor. So as the sediment comes out, its images are taken, and this is a composite. And the 340 meters beneath the seafloor is approximately 75 million years old. So this is a whole lot of Earth's history, epochs we're talking about. And so the one other thing about this is cores are taken out in a series. They're not just one big long chunk coming up. Sediment is drilled and comes up, and each core is approximately 9.5 meters long, and then they're subdivided. So that particular section of sediment is the 9th core, and then you would have it subdivided into different sections. So, and then another important part is the sediment conditions. So, like I said, this is very different than we would consider normal microbial conditions, where when we experience microorganisms, it's usually an overgrowth. They just grow and become a big problem. But again, these conditions are extremely limited. So there's no photosynthesis, and any carbon that is there was buried at the initial time of sedimentation. It's estimated that 84% of the microorganisms in seafloor sediments survive on energy that less than previously thought possible for survival. So just no energy for anything. And so this leads to doubling times of possibly thousands of years or at all. So even for those sediment samples that are millions of years old, you're looking at organisms that were are very closely related to the organism at the time of burial, because they just don't have the energy to divide and grow. So when a cell enters the marine sediment, it has three paths to possibilities. It can be metabolically active, and that allows for the possibility of DNA repair, and it also for the possibility of division. However, it takes energy. Dormant is the possibility of being reawakened if conditions are right. However, there's no DNA repair that occurs, and so it will eventually die, and that's the third state. A lot of cells just die. But like I said, the DNA can survive for potentially thousands of years. And so even those cells that can adapt to low energy environments will eventually die when they reach the sterilization limits of very low energy or high temperatures towards reaching the very bottom of the rock at the bottom. So we were looking at a very particular type of DNA degradation to compare samples. And so we're looking at uracil substitution. So this is a process of deamination. And so at the green box, you can see where there's an amine group, and that attaches from the cytosine nucleic acid, and then it turns into a uracil. And so that is identifiable during DNA sequencing. And so there's an enzyme called uracil and glycosylase that produces a cut in the DNA strand at a uracil site. So it does occur naturally, but it can also be used in a laboratory to remove degraded DNA that had a lot of uracil substitutions. And so again, our paired samples, we had untreated samples that are a mixture of the active DNA, living DNA, and then the degraded DNA, the DNA that has a lot of uracil substitutions. And then we compared that to a treated sample, which just contained metabolically active DNA. So here are the methods. So this, as John said, it was a process of several years and a lot of effort from different people. The original sample locations were in the Bering Sea in 2009 and the Indian Ocean in 2006. And the samples were preserved at -80°C and brought to an onshore laboratory, the University of Rhode Island, where John Kirkpatrick processed the samples for DNA analysis. Once the analysis was complete, the information was uploaded to the Visualization and Analysis of Microbial Population Structures database, or MES, that houses a lot of environmental DNA data. And then I conducted the analysis interpretation. But this infographic is really great at showing just how much effort is required to process these samples. So here is the results of our bacterial analysis. The Bering Sea had a total of just over 3 million reads, as did the Indian Ocean. And so reads are just the number of DNA sequences that were identifiable. And so we had a total of 714 taxa, or different organism types, in the Bering Sea, and 879 in the Indian Ocean. So a little bit more variety of microorganisms in the Indian Ocean, of bacteria. And so this is an example of the type of analysis that we conducted. So again, these are paired samples. The reads for UNG positive are from the same sample as the reads for UNG negative, which is, again, the UNG positive is treated, so active DNA. UNG negative is all the DNA, degraded and active. And so we normalize these results to exclude any taxa that only had a couple of reads or was not deemed significant. And so normalization was done by taking the number of reads for that particular organism and dividing it by the total reads for that sample. And then the normalized treated and untreated samples were added together, so we got the sum normalized, and any taxa that had a number greater than 0.01 was included in the results. Anything less than that was deemed not significant because there wasn't enough reads. And then we created a ratio, and this ratio is really what we're going for. So we divided the untreated normalized sample by the treated normalized sample, and we get a ratio of degradation. So in the top row, we get 13.284. That's our ratio. That means there's a lot more degraded DNA in that sample than there is active DNA. So we know that organisms are dying at that sample. So here are our results. And so this is a combination of both the Indian Ocean and the Bering Sea data. And the x-axis, again, it starts in the upper corner at 0, that's the seafloor, and then it goes down into the sediment. So we're seeing age as we go down. And then the overall number of taxa is the x-axis. So we see a lot more taxa in the Indian Ocean at the very top. And then we see a gradual decline in all of the taxa as the age increases. Also note that the darker colors are the untreated samples or the degraded DNA, and there's a greater number of degraded taxa at most of the locations, and then the active DNA. And we'd expect to see that because we expect the different types to be dying as depth increases, as age increases. So this is a graph of our Bering Sea taxa degradation as depth increases. So again, on our y-axis, we have depth. The upper corner is as depth is the seafloor, and then depth increasing. And then the x-axis is a little different. It's the number of degraded taxa. So it's not the overall number of taxa. So towards the seafloor, we have 4 taxa that are dying rapidly. And then so again, we see this die off towards the seafloor, and then around 10 meters, we see a stabilization of the microbial populations. And then we have a second major die off towards as depth increases. And this is where microorganisms that were able to adapt to the sediment are unfortunately succumbing to the low energy environment and can no longer continue to survive. So we did have our keel data as well, but however, the reference library was not precise enough at the genus level and didn't give us clear results. So a possible future project could be a de novo clustering that just looks at percent relatedness. We can also potentially look at the evolutionary history of one organism that's present through an entire core. Because again, the microorganism that is thousands of millions of years old is really closely related to what it was at the time of burial. So you can see little changes as you go up towards the seafloor and see the whole evolutionary history. Also, you can see shifts in microbial communities at different climatic events or potentially large volcanic events, and see how the Earth's climate and environment affected the communities in the seafloor. So conclusion is, yes, we did see a pattern of degradation within our 2 sample locations and also in the literature. And so placing these microbial communities in the context of the larger history and the larger contents of the sediments, because they're not just looking for microorganisms during these research cruises, they look for, nutrients and paleo fossils and all these other things. And so placing that into a larger picture lets us see how these communities react to past Earth climate, how they could potentially react to current climate changes and in the future. So it's really important to understand what's happening in the seafloor, because we have this wonderful time capsule of the past. So, any questions?